Okay, so today we're going to talk about the rest of chapter 24, which is the digestive system. And we're going to start with the pancreas, then we're going to look at the liver, then we're going to look at the small intestine and the large intestine, and the defecation reflex. And then we'll talk a little bit about these macromolecules that are getting digested. So first, let's start with the pancreas. So the pancreas is a gland that provides digestive enzymes and buffers to neutralize the chyme that is leaving the stomach and going into the small intestine. The pancreas is just behind the stomach, and it's a little lateral from the duodenum, and it fits right inside that curve of the duodenum pretty nicely. It just snuggles right in there. It is a pinkish gray gland that's about 6 inches long. It has a head, and it has a body, and it has a tail. It also has a large pancreatic duct that delivers secretions from the pancreas into the duodenum. The pancreatic duct meets up with the bile duct that's coming from the liver and the gallbladder. Together they form a common duodenal papilla that empties into the duodenum. The pancreas is both an endocrine organ and an exocrine organ. So let me explain what that means. The endocrine glands are going to secrete their secretions back into the body. So those secretions are going to end up going back into the bloodstream. That's what means endocrine. Endo meaning going back in. So the endocrine responsibility of the pancreas is to secrete insulin and glucagon into the bloodstream. Insulin and glucagon control blood sugars. The exocrine function of the pancreas is to secrete a pancreatic juice. So the pancreatic juice consists of an alkaline mixture of digestive enzymes, water, and ions. The enzymes are going to break down ingested materials to be absorbed, and then there's also water and ions that are being secreted to dilute and neutralize the acids in the chyme. So we'll talk about all of that too. The pancreas secretes about 1 quart of pancreatic juice every day. It's regulated by hormones that are secreted from the duodenum. So when the duodenum releases the hormone secretin, that causes the pancreas to release a watery, buffery solution into that pancreatic duct, which will then deposit into the duodenum. Another hormone from the duodenum called the CCK or cholecystokinin, when that's released from the duodenum, it causes the pancreas to secrete all these pancreatic enzymes. Those pancreatic enzymes then will move into that pancreatic duct, and they as well will be deposited back into the duodenum. A disorder called pancreatitis is a disorder that is an inflammation of the pancreas. It's an extremely painful inflammation of the pancreas. Pancreatitis can be caused by a blockage of the duct. It can be caused by a bacterial or viral infection. It can be caused by ischemia if there's not enough blood supply going into the pancreas. And it can also be caused from drug reactions, especially alcohol. Speaking of the blood supply, the pancreas is supplied by several different arteries, including the splenic artery and the superior mesenteric artery and the common hepatic arteries. And it is drained by the splenic vein. Let's talk about the pancreatic enzymes. There's pancreatic amylase, pancreatic lipase, pancreatic nuclease, and pancreatic protease. So we'll start by looking at amylase. Amylase is an alpha amylase. It breaks down starches into monosaccharides. So it's breaking down complex carbohydrates into simple sugars like glucose. and it is identical to the salivary amylase. So if you remember, salivary amylase stopped working in the stomach because it was too acidic. So carbohydrate digestion stops in the stomach. But once amylase is secreted into the duodenum, then the digestion of these starches and polysaccharides begins again. There's also lipase, pancreatic lipase. This is going to break down the complex lipids into fatty acids. Then we have nucleases. Nucleases break down RNA and DNA into nucleic acids, which are a smaller monomer. And then finally we have the proteases. Now the proteases make up 70% of all the pancreatic enzymes. They break down large proteins and peptidases, and they break them down into smaller peptide chains, which can then be broken down into amino acids. And then amino acids can get absorbed in the intestines. The proteases are first secreted as an inactive proenzyme. So there's several different proteases. The proteases are secreted as inactive proenzymes. So here are the inactive proenzymes. We'll write those on this side. And over here then we'll write the activated proteases. Okay, so the trypsinogen is an inactive proenzyme. It will become activated to trypsin. So trypsin is a protease. And chymotrypsinogen will become activated to chymotrypsin. Then we have carboxyl peptidase, which becomes activated to carboxypeptidase. And then finally we have proelastase, which becomes activated to elastase. Now all these proteases will break the peptide bonds in polypeptides to make them smaller, and they become dipeptides, where there's only 2 amino acids, or tripeptides, where there's only 3 amino acids, or even break them down as small as just single amino acids. Next, we're going to look at the liver. The liver is the largest visceral organ. It's divided into 4 different lobes. It's a firm reddish-brown organ that weighs about 3.3 pounds, and it's found in the right hypochondriac and epigastric regions. So the stomach's on the left, and then the liver's on the right, underneath the rib cage. The falciform ligament is a ligament that divides the liver into a right lobe and a left lobe. Then there's a round ligament, and the round ligament is a thickening of the falciform ligament, and it's on the posterior side of the liver. Another name for the round ligament is ligamentum teres, like this. And the round ligament was the path of the fetal umbilical vein. And then after the baby was born, it became the round ligament or ligamentum teres. On the back side of the liver, we can see the left lobe and the right lobe, but then we can also see the other 2 lobes called the caudate lobe and the quadrate lobe. The caudate lobe is more superior and the quadrate lobe is more inferior. The liver receives blood supply through the hepatic artery proper. And this hepatic artery proper is a branch off of the abdominal aorta. The liver also receives blood supply from a vein, and this vein is called the hepatic portal vein, right here. And the hepatic portal vein is coming from the intestines, from the small intestines, and they're bringing nutrients to the liver so the liver can decide what to do with those nutrients. The blood from the liver then drains into the hepatic vein. The hepatic vein then drains into the inferior vena cava and heads back up towards the heart. There are a couple conditions of the liver that I'd like you to be aware of. The first one is called cirrhosis. Cirrhosis of the liver is when fibrous tissue replaces the normal hepatic tissue. Another condition of the liver I want you to know is hepatitis. And hepatitis is caused by viral infections. When the liver is damaged, it actually has a high capacity to regenerate as long as there's normal vascular patterns, so there's normal blood supply. But if it doesn't have the ability to regenerate, then a person might need a liver transplant. A liver transplant success rate is going to be higher with younger people than it is with older people. Or if a liver transplant isn't available, liver dialysis could also be used. Liver dialysis, or ELAD, which stands for extracorporeal liver assist device, is used in the case of liver failure or chronic liver disease while a person is waiting for a liver transplant. I want to talk to you about the hepatic portal system. The hepatic portal system is where the hepatic portal vein, which is right here, meets up with the hepatic artery, which is right here. So if you remember, the hepatic artery is a branch off of the aorta, and the hepatic portal vein is where the superior mesenteric and the inferior mesenteric combine to become the hepatic portal vein. And then the hepatic portal vein meets up with the hepatic artery, and they become the hepatic portal system. The hepatic portal system is where that artery, where they come together, starts to branch off into smaller and smaller vessels and become these sinusoids, or these sinusoidal capillaries. The sinusoidal capillaries, if you remember, they have holes or pores in between each of the endothelial cells that are lining those blood vessels. So those are big gaps in the capillaries where things like plasma proteins can enter into the blood. Those sinusoidal capillaries then pass by the cells of the liver, which are called hepatocytes. The hepatocytes are arranged like the spokes of a wheel, and they're producing things like plasma proteins, and they are deciding what to do with the glucose that's coming to them from the intestines and what to do with the amino acids. So they've got all of these functions that they have to do to make decisions about what do they do with the nutrients that you're receiving from the intestines. And then the blood from there moves into a central vein, and then that central vein will merge together with other central veins, and they will then exit the liver through the hepatic vein. So this merging of these blood vessels and then the passing by of the hepatocytes is called the hepatic portal system. The hepatic vein then will merge into the inferior vena cava and head back towards the heart. The pressures in the hepatic portal system are very low. They're around 10 millimeters of mercury or so. You don't want a lot of pressure in these sinusoidal capillaries that have these big gaps in them, because otherwise fluid would move out of these sinusoidal capillaries into the peritoneum, and then fluid would build up, and that's called ascites. A rise in this portal pressure is called portal hypertension. It distends the capillaries and the veins as well, and they could actually rupture. And if they rupture, then they cause extensive bleeding. Hepatitis and alcoholism can be a cause of portal hypertension because they cause constriction of these capillaries, and that increases the pressure. The liver has 200 functions overall, but they all fall under 3 different categories. There's a metabolic function, a hematologic function, and then there's bile production. So let's first talk about the metabolic function. All blood leaving the intestines after absorption goes to the liver through the hepatic portal system. The liver then extracts the nutrients or toxins if those were ingested, and then they decide what to do with those nutrients. They release some nutrients into the blood and they store any excess nutrients. They also mobilize the nutrient reserves whenever that's needed if there isn't enough nutrients in the blood. So let's look at carbohydrates. The liver stabilizes the blood glucose at 90 milligrams per deciliter. So the liver is a very important part in keeping your blood glucose levels normal at 90 milligrams per deciliter. If the blood glucose levels are over 90, then the hepatocytes pull the glucose from the blood and they store it as glycogen. Or to make lipids, which can be stored in the liver and other cells. If the carbohydrates in the blood is less than 90 milligrams per deciliter as it comes up to the liver, then the hepatocytes break down glycogen into glucose. So then the glucose can be put back into the blood and then the cells can use the glucose. Or the liver has the ability to make new glucose from other carbohydrates or amino acids, and that process is called gluconeogenesis. The second metabolic function is lipid metabolism. If triglycerides, fatty acids, or cholesterol levels go down or decline, then the liver will break down lipid reserves and release those products into the blood. If lipid levels are high, then the liver will remove them and store them. So the liver is trying to decide, should the liver remove the lipids and store them if there's excess, or should the lipids be released into the blood? Those are the decisions that the liver has to make. Lipids aren't generally brought to the liver, though, through the hepatic portal system. At the intestines, when lipids are absorbed, instead of going into the bloodstream, they go into the lymphatics. And then the lymphatics takes them around the body for a turn, and then it goes into the liver. And at that point, then, the liver will decide what to do with the lipids. So those lipids are coming from the general circulation. Then the liver has the amino acid metabolism responsibility. So same thing with the amino acids. Any excess amino acids will be removed and stored, and they will be used to make proteins. Or they'll be converted to lipids or glucose, so they can be used as energy. The fourth metabolic function of the liver is toxin removal. So one of the toxins in your body that's produced is called ammonia. When the liver breaks down proteins into amino acids, it does that through a process that's called deamination. And so proteins are made up of just lines of amino acids that are connected together by peptide bonds. And when these amino acids get popped off, they produce a toxin that's called ammonia. And ammonia is, because it's toxic to the body, the liver has to neutralize the ammonia by converting it to urea. Urea is harmless and it can be excreted by the kidneys. The liver basically has a couple of options with toxins. It can either store them right in the liver, which makes the liver toxic. It can inactivate them, or it can excrete them in the bile, which would end up in the feces. Another metabolic function of the liver is vitamin storage. So the liver can store fat-soluble vitamins like A, D, E, and K. Those are all fat-soluble vitamins, as well as vitamin B12. They're stored in the liver and then they'll be used as needed. Another metabolic function of the liver is mineral storage. The liver will convert iron reserves, for example, into ferritin, and then it will store ferritin. And then the last metabolic function I want to mention is called drug inactivation. So the liver removes and breaks down any drugs that are circulating in the blood, and that limits the duration for how long that drug can affect the body. Whenever giving a person a drug, you have to take into account the rate at which the liver will remove that particular drug from the bloodstream. For example, a drug that's absorbed quickly must be administered every few hours to keep the blood concentration at a therapeutic level. Otherwise, it will be taken to the liver right away and it will be broken down and inactivated. Next, let's talk about the hematologic regulation that the liver has. Hematologic regulation is basically saying how does the liver regulate the composition of the blood. Well, the liver receives 25% of the cardiac output. So the liver ends up being the largest blood reservoir in the body because it's receiving a lot of the blood that the heart is pumping out. Then as the blood passes through the liver, the following things are going to occur. So first of all, phagocytosis occurs. The macrophages in the liver are called stellate macrophages, or you might hear them as Kupffer macrophages, which is what they used to be called. And they engulf anything that looks abnormal. They are going to engulf things like old and damaged red blood cells or cell debris or pathogens that are in the blood, and they then present these antigens on the surface. And they wait for those T cells to come by and inspect them and see how dangerous they really are. So phagocytosis and antigen presentation is one of the ways that the liver monitors the blood. The second hematologic function of the blood is that it makes plasma proteins. The hepatocytes in the liver make and release most of the plasma proteins. These plasma proteins include things like albumins and other transport proteins and the clotting proteins like fibrinogens. And the complement proteins, which are part of the immune system. The next hematologic function of the liver is that it removes circulating hormones. The liver removes and recycles hormones like epinephrine, norepinephrine, insulin, thyroid, and then some steroid hormones like estrogens, progesterones, testosterone. So these hormones really don't have a long life. As soon as they pass through the liver is going to remove them from the blood. The liver also absorbs vitamin D3. It absorbs it from the blood and it converts it to an intermediary product that is then released back into the blood, and it is absorbed by the kidneys then, and the kidneys then will make calcitriol with it. Another hematologic function of the liver is that it removes antibodies. From the blood. So it absorbs and it breaks down these antibodies into amino acids. And the last hematologic function of the liver is that it removes and stores toxins. So we saw this in the metabolic function. I'm mentioning it again here in the hematologic function because they kind of are similar. And so it removes these toxins and it will store them in lipid deposits so that they don't disrupt other body cells. An example of this would be like the insecticide called DDT. Toxins that aren't stored are going to be broken down or excreted in bile. The last function that I want to mention of the liver is the production of bile. So the liver makes bile and excretes it into the duodenum. Bile is made up of water and ions and bilirubin. Bilirubin comes from hemoglobin from broken down red blood cells. It also contains cholesterol and it contains lipids that are called bile salts. The bile salts are made from cholesterol. Bile salts break down large lipid droplets and they break it apart so the lipids inside can be broken down by pancreatic lipase. Let's take a look at this picture so you can see what I mean with the bile salts. So here we have these large lipids right here. All of these are large lipid droplets. And those droplets are going to be broken apart by the lipids so that the lipids inside can be broken down by pancreatic lipase. So the pancreatic lipase can only really get to these lipids if they're individual lipids. So we need the bile salts to break apart this great big molecule of lipids and break it down into these individual lipids so that the pancreatic lipase can then break down these lipids into triglycerides, which can then be absorbed. This process is called emulsification, where the bile salts break down those larger lipids into smaller lipids. The bile salts also promote absorption of lipids in the intestine. So when the liver produces the bile salts are secreted into the intestines. Once in the intestines, the bile salts can promote the absorption of lipids in the intestine, and then they're going to be reabsorbed themselves and recycled and returned to the liver. That cycling of bile salts from the liver to the small intestine and then back to the liver again is called enterohepatic circulation. All right, let's look at the gallbladder. The gallbladder is a hollow, pear-shaped, muscular sac. It has a fundus, a body, and a neck. It's connected to a cystic duct. That cystic duct meets up with a duct from the liver that's called the common hepatic duct. And then together, those two ducts merge to form the common bile duct. The common bile duct then meets up with the pancreatic duct from the pancreas at an area that's called the duodenal papilla, and then that opens up into the duodenum. The gallbladder stores and modifies bile. So the liver produces the bile moves into the common hepatic duct and then into the gallbladder where it can be stored and modified. The bile will be released into the duodenum, but only when the duodenum produces CCK. So CCK causes the release of bile into the duodenum, and CCK is produced by the duodenum. When CCK is released, the sphincter relaxes around the duodenal papilla opens up, and the contractions of the gallbladder force the bile into the duodenum. The amount of CCK increases markedly when chyme coming into the duodenum contains a large amount of lipids. So more bile will be secreted if the chyme has more lipid content in it. When the gallbladder is full, it will contain anywhere from 40 to 70 mils of bile. The composition of bile changes in the gallbladder. So once the bile is in the gallbladder, it's going to be modified. Water gets absorbed and the bile salts become concentrated. If it becomes too concentrated, then crystals can form, and that's called cholecystitis. Cholecystitis is when the stones are too large and they damage the gallbladder walls and they block the ducts. In that case, the gallbladder would have to be removed. During removal, bile production is still normal because the liver is still producing bile, but instead of the bile being stored in the gallbladder, it goes directly into the duodenum. So it didn't have the gallbladder to concentrate it, and the bile will be dilute, and it won't be timed as well with the entry of food. Okay, I think that's all we're going to do for this video. We're going to start again in the next video talking about the small intestine. You guys can take a break. I'll take a break too, and then we'll come back together in the next video.